So good afternoon. Oh, come on. It's too cold out here for us to have cold voices. Good afternoon. All right. We bring you to the 29th annual Dr. Martin, Reverend Dr. Martin Luther King Jr. Health Sciences Program. It is of the colleges and units that are the health science schools. And that's important. Let me just pause there because most times folk don't know that the health sciences collaborate for over 29 years. And we thank the deans and the directors for supporting this event for all of these years. And so I am Dr. Patricia Coleman Burns, and the way we do it in the health sciences is each of us sort of step forward every year and say, oh, our unit will take on this work. I don't know why we do this. It is an amazing amount of work. I want to acknowledge the people that are viewing this from wherever they might be. We know there are what we call watch parties all over the campus, some individually and some in different units. We welcome you, and if you could just give them a big hand here in the present audience. We know you're there, we hear you, and we respect you. So the brief history is what I've already given you. We do this every year. I should be very careful to say who's going to do it next year. I saw a smile. I saw public health. Okay. But so kinesiology or public health, someone will pass the baton on to. So what this means is that if you look at the years that the university has been celebrating Dr. King, you will notice the health science deans and came right up immediately to do their part. So here we are. Why don't you just take a moment and see all of the health science schools and colleges and units that support this event. Give them a hand. And if you don't see your unit and you want to join us, please feel free to. So right now, this is a dean's event. If all the deans of the health science schools and colleges would please stand and let us just say thank you very much for your continued effort. There's another slide that will run that has their pictures, and you'll be able to see and to contact them. The other thing is it takes, when I asked Guy, who is somewhere running around, I said, Guy, we need to list all the people on the team. He said, Doctor, they call me PCB, there are 40 people who make this possible. So with all of the health science MLK Planning Committee, please stand and be recognized. They do so much work. We started this year with Childish Gambino as a viewing and a program, and we're looking forward to an event on March the 20th that I'll tell you about whenever the slide comes up or later. I do have to acknowledge it takes a real village to do this kind of event. The food, the coordination, the speaker, and in addition to the planning committee, we have in our units the staff, and I do want to recognize these folk. So give them a hand. There's Guy. Guy, stand up so people, stand up, Guy, so they can see your face, is our main person. So you received when you came in a survey. In order to know what we're doing well, what we need to do better, you've got to respond. But also, we have a raffle. And the code word for those online is Dr. Taylor, unravel disparities. But for those of you in the audience, just sign at the bottom on your way out at either one of the doors, tear off, and you will be entered into a raffle, and we will do that immediately following this event. And the raffles, the gift cards are from $20 down to $10. And so there are those opportunities. That's my job. I can now sit down. Thank you very much. Let me introduce to you my dean, Dean Patricia Hearn.
Thank you, PCB. So this really is a marvelous event, and I get to do the best thing of all, which is introduce a really phenomenal woman that I think you're going to enjoy learning from today. So let me tell you a little about her. Dr. Jacquelyn Taylor is currently the Ferguson Endowed Professor in Health Equity at the New York University, and also an affiliated professor at the School of Medicine. She has a long history. She took her undergraduate degree, master's degree, and PhD all from Wayne State. We won't hold that against her. That's a very fine place. But what you read as you go back through her background is that she became interested in African American women and their health in particular needs and what makes them unique from the time she was actually a very young person. And I think that's why she's been able to move to the place of prominence that she is today. So in addition to those titles, I think it's important you know several other things about her. She's a fellow of the American Academy of Nursing, but because of her research, she covers many different disciplines: medicine, basic science, genetics. Her work really focuses on the genomics of chronic disease in African American populations. She's been engaged in women's health, but many other things as well. Her research examines the effects of psychological, genetic, and epigenetic, very important, epigenetic factors on blood pressure in Black African American women and in their young children. What you don't know is that she started her career as a pediatric nurse practitioner. And for those of us in the audience who are clinicians, you realize that you never really leave that behind you. It laces our intellectual pursuits and our research without one bit of doubt. She has a variety of awards, but I did want to pick out particularly the Presidential Early Career Award for Scientists and Engineers, PCASE, which is actually the highest honor bestowed by the U.S. government to outstanding scientists and engineers in the early stages of their research careers. So that's really an outstanding accomplishment, even at a very young age. I understand her long-term goal is to develop nursing interventions to reduce and prevent omic environmental risks associated with health disparities. I think her challenge today, and one which I know she is very capable of doing, will be to teach us something about the genetics and genomics of hypertension in African American women. Dr. Taylor, this is all yours. Welcome. Let's have a hand for her.
Right. Thank you so much for that wonderful introduction. I hope I live up to all the kind words that you just said about me. So again, it's always a great pleasure to be back here in Ann Arbor. I started my academic career, my first tenure track position here at the University of Michigan. So today, what I would like to talk to you about are some of the things that I did early on in my career to build up the research my research platform that I currently have now that focuses on genomic and environmental factors to unravel health disparities for hypertension in Black women. So just as an introduction, I know Dr. Hearn just told you a little bit about my educational background, but I like to always tell this story when I tell people how I got started. I started off working in a laboratory at Wayne State University in undergrad, and the way that I was introduced to the laboratory, I went to the student services office. I was just looking for a job, poor college student. You know, I really need to just get a job so I can continue to pay tuition and so on. And so I talked to the guidance counselor, and they said, what are you really interested in? And I said, I'm interested in hypertension. This is a problem in my community, and you know, people in my family have high blood pressure. I want to know more about how the body works and the physiology behind what, you know, how people become ill and what we can do to make sure that they can get better. And this person was really tired of hearing what I had to say. But at the time, I was 18 years old, and I didn't realize that she was exasperated with me. And she said, well, you need to go and talk to the chair of the physiology department. And I said, okay, who is that? So I didn't realize that you should probably make an appointment, or that the chair of the physiology department was probably an important administrative role. I was 18 years old and just got, stepped foot on campus. So I went to talk to Dr. Joseph Dunbar, who was the chair of the physiology department at the time at the medical school at Wayne State University. And, I could hear, like, on the other side, I talked to his assistant, and, I said, y'all wanted to talk to Dr. Dunbar about, all these ideas I had. And I could just imagine on the other side of that telephone, like, who is this person that just showed up to my office? So I feel like he just took pity on me. And, he had a R25, which is a grant to train undergraduate minority students through the Minority Biomedical Research Program. And so I was able to obtain a position and work in one of the laboratories that was a part of the R25 program. And I did that for the duration of the time that I was an undergraduate student. So I made myself useful. I learned how to use every piece of equipment in the laboratory. I did all types of experiments with rats, and did everything that other laboratory assistants did in the lab. So then I continued on at Wayne State to pursue my master's degree. I became a pediatric nurse practitioner, and they also had a school nurse practitioner program at the time, so I was able to complete that. I was also encouraged by my advisor to continue with the basic science knowledge that I had and to apply to the Summer Genetics Institute, which was sponsored by the National Institute of Nursing Research. And so I was fortunate enough to be accepted into that program, and spent 2 months in Washington, D.C. Doing work there. And then I continued at Wayne State to complete my PhD, where I looked at parenting attitudes and lifestyle behaviors on hypertension among African American parents and grandparents. My master's advisor that encouraged me to go to the Summer Genetics Institute was Dr. Chandis Covington. My PhD advisor was Dr. Olivia Washington, who had expertise in aging and urban health aging. My master's advisor had expertise in pediatrics. I also continued to do a postdoc in urban health aging because in genetics, you really want to look at family pedigrees, and I felt very comfortable with my knowledge in pediatric populations, but I felt like I needed more training in the older population, other side of the continuum. So I applied, and my mentors for my postdoc were Dr. Peter Lichtenberg and Dr. James Jackson, who, of course, is here at the University of Michigan. As I continued to do work in my postdoc, where I wanted to look at 3 generations of genomics of hypertension in 3 generations of African American women, I quickly learned that my statistical knowledge was not as advanced as it needed to be in order to conduct the analyses that I needed to do to answer the research questions that I had. So I applied to Washington University in St. Louis through a PRIDE program, another, I think, R25, and was accepted as the first nurse into that program. I remember calling up the person that was running the program because nurses were not included in the people that were eligible for this program. It said, biostats, medicine, all the other health professions, but not nursing. So I called and I said, well, I'm a nurse scientist, but I'd be eligible for this program. And they said, sure, apply. So Dr. D.C. Rao, who is a epidemiologist, and Dr. Victor Devia-Roman, who is a cardiologist, were my mentors in that program. Now, I put all the pictures of all of my mentors here because I think this makes a strong point in why I do the work that I do and why I think it's important in unraveling health disparities. As you can see here, I have people from various disciplines: social science, a physician, epidemiologists, gerontologists, nurses, and physiologists. These were the people that trained me to do the work that I do. It never occurred to me that I only needed to work with nurse scientists. It really was ingrained in the type of training that I received to always collaborate cross-disciplinary. In addition to that, my mentors were from various backgrounds. So not only did I have the great benefit of having mentors that were from various specialties, but they came from various ethnic, racial backgrounds, men and women. So I just hope that more people have this the great fortune of having excellent mentors from various backgrounds and racial ethnic groups. So a little bit about my work background. I've worked as an RN at Mercy Hospital here in Detroit. I had a mobile clinic where I was a pediatric nurse practitioner, and we served underserved communities in Southwest Detroit and the east side of Detroit. And we took our mobile clinic to the schools, and we provided all the health care to those families. Now, I did everything, including driving the van. These are some Detroit Lions players because the mobile clinic was funded by Edsel Ford. And so one of the schools was the Detroit Lions Academy that we would service. I don't know who any of these players are, but I'm sure they're excellent players. But so I did a lot of clinical work, and then I was fortunate enough to obtain my first tenure track faculty position here at the University of Michigan as assistant professor. And my very first dean, who is in the audience today, was Dr. Ada Sue Henshaw, and she continues to be a great resource and mentor to me even to this day. Even the other day, just talking to her about career advice and anything that research advice, anything that has to do with academia, she is a wealth of knowledge. So I always turn to her to steer me in the right direction. Another great mentor that I have here at the University of Michigan is Dr. Sharon Cardia, who's at the School of Public Health. She was actually my primary advisor on my first Career Development Award, K Award, that was funded here at the University of Michigan. And we still continue to work together to this day, many, many years. I won't date myself, many years later. And then I also spent time as a visiting professor at Massachusetts Institute of Technology in their biology department because I do work in whole genome sequencing. But they had an interest in how to translate the work that they do in basic sciences to usefulness in clinical practice. And that's what nurses do. So I spent 2 years as a visiting professor working with Dr. David Hausman in the biology department at MIT. And I was the very first nurse ever selected to be a visiting professor through their Martin Luther King Jr. Visiting professor program. And so now, more recently, I've been at New York University as an endowed chair, the Vernice Ferguson Endowed Chair of Health Equity for the past year. And this is my new dean, Dr. Eileen Sullivan-Marks, of which I'm sure she will become a mentor in many ways. She's just not aware yet. But so I just wanted to give you that little bit of background so that you will understand why I'm here to talk to you about this today and how it really relates to the things that I think are important to unraveling health disparities among Black populations. And I'm going to talk to you about some of the pilot work that I've actually done here in Michigan and how I built on that work and how more questions arose and how I worked to continue to unravel the even more questions that continue to arise as we continue to do this work. So because this is the health sciences, I won't explain DNA to you. I think you understand it very well. You know about polymorphisms and single nucleotide polymorphism SNPs, and you know about base pairs. So I'll just go right into the very first study that I did here in Detroit. So as a postdoc and during my first academic position here at the University of Michigan, I started with a study where I wanted to look at 3 generations of African American women and look at genetic risk for hypertension. Now, studies had been done already looking at genetic risk for hypertension in various populations, although most of those studies did focus on Caucasian populations. And the thing that was lacking in the studies that had been done in minority populations were a lot of them did not have pediatric populations included in these studies. So we had the Jackson Heart study. We had a lot of studies with sibling pairs or single adults, but we did not have a lot of studies that focused on looking at identifying genetic risks early in life prior to phenotypic expression of the disease. So why look at hypertension? And why look at hypertension in African American women? and particularly genomics of hypertension. Well, we know that African American women have the highest incidence and prevalence of hypertension of any ethnic group and of any gender group in the United States. We also know that African Americans develop hypertension at an earlier age. At the time, Detroit had the second highest rate of hypertension in the United States, just behind Memphis. The other things that we knew that lifestyle behaviors played a huge role in hypertension, and we also knew that genetic polymorphism or genetic inheritance played a role. So I wanted to look at if we could identify this early in life, and if we could, then that would open the door to potential interventions early on. So with a P20 pilot from the National Institute on Aging, and then following up with the K Award that was funded here at the University of Michigan, I was able to start this small pilot looking at 3-generation pedigrees. And it was a small, just descriptive study looking at a few SNPs. It wasn't a large GWAS because at the time we weren't doing large GWASs. But I wanted to see if this was something that we could identify in childhood. So the SNPs that we looked at, we wanted to look at the ones that had already been identified in the literature to be risk factors for hypertension in blacks. But I wanted to, and so all the sodium bicarbonate transport gene, so the SLC4A5 gene, which is located on chromosome 2, had been identified as such. And there were 4 SNPs that we were going to look at on that particular gene. So very simple, just 4 SNPs, small pilot study. Descriptive research design, 3-generation families, grandmothers, mothers, and granddaughters. That's it. This was the eligibility criteria that we used. And I always point this out because when we first started this study, we did not require that all 3 people be at the same place at the same time. But then I quickly realized that as I was driving from like the northwest side of Detroit to the southwest side of Detroit to east side of Detroit, that this was just taking way too much time and manpower to track all these people down and to collect the data. And so what I did is I would just contact the grandmother. And when I contacted the grandmother, I never had any problem with the other 2 generations. She said, yes, baby, I'll make sure that they're going to be right there at 3 o'clock, and they would be there. So I never had to worry about a thing. So that was great. Certain recruitment strategies that we utilized the University of Michigan Women's Health Registry. We utilized the Wayne State University Institute of Gerontology Participant Resource Pool. I posted flyers at beauty salons, grocery stores, and so on. And then it dawned on me after we started the study, I'm a member of a historically black sorority, and I go to these sorority meetings every month, and I realized, oh, there's multiple generations of African Americans at this meeting, African American women at this meeting every single month. Why didn't I think about this before? So I, reached out to the president of our chapter, and I was able to recruit from Alpha Kappa Alpha sorority, of which I was a member of. And then we also utilized social networks. So once people started to participate in the study, they were very interested. They would tell their neighbors and their friends, and they would contact us to take part in the study. This was interesting because when I first started out in the study, I was told that, well, you'll never be able to get black women to participate in the genetic study because of the lack of trust. We actually had more people that wanted to participate than we could enroll due to our budgetary constraints with the pilot grant. And so I thought it was important to publish this so that people could see, not only were we successful in recruiting and retaining people in the study, but these were the methods that we used that helped to make our strategies very successful. These were the measures, some of the measures that we had in the study. For this talk, I'm going to talk to you today about 24-hour food recall. I thought this would be one of the most difficult variables to collect because I thought, well, you'll just get socially desirable answers. I ate fruits and vegetables all day and drank water. But people were very honest with what they ate. If they had McDonald's, if they had, something that wasn't necessarily healthy for them, they would still be very honest and tell you what they ate. The other thing was that if they made something that was homemade, they would tell you all the ingredients that they used. So I got some great recipes. It's like, that's how you do that. So this, from this triad, you can see here that this was the rates of hypertension among this group. And as you can see, it's well above the national average of 42% among African Americans. So the grandmothers, 73%, had previously been diagnosed with hypertension. Mothers, 42%, and even the granddaughters, 12%, already diagnosed with hypertension. If you look at the blood pressures here, and this was based on JNC7 guidelines and the old guidelines for hypertension, if we use the guidelines for today, even more people in the study would fall into the stage 1 hypertension category. But a lot of the people that had been diagnosed with hypertension, their blood pressures were still in the stage 1 hypertension range. For the grandmothers, 146/82, and then the mothers, 134/83. But a lot of these people were also taking antihypertensive medication. So you would think that more people would have better control of the blood pressure, but not so much. We also looked at body mass index, and what you can see here is that all 3 generations fell into the overweight or obese category. All 3, even the granddaughters. And then we also looked at percent body fat, and among all 3 generations, all 3 fell outside the normal limits for percent body fat for women. When we just did a simple correlation analysis for blood pressure and each one of the polymorphisms, we saw what we really expected. We thought that they would be significant because they had been shown to be significant in other studies. And we published this in Biological Research for Nursing. But then we used our, when we wanted to look for the interaction with sodium intake, this was the analysis plan that we used. And as you can see here, it's not as simple as just a correlation or a simple regression, right? So we had to conduct a multivariate linear regression, linear mixed models, multivariate mixed models for SNP and sodium intake interaction. And this is why I had to do the additional training in advanced cardiovascular data analysis. And I also know that I need to work with people in the School of Public Health that really know what they're doing, like Sharon Cardia's group and Jennifer Smith and Aaron Ware at the School of Public Health. So we did all of these analyses, correcting for antihypertensive medication use. And once we did that, only one of the 4 SNPs remained significant for interaction with dietary sodium. And the most interesting thing about this was that the T allele was protective against increases in systolic blood pressure, even when people reported consuming more sodium than the daily recommended allowance. So this gets to the unraveling, like, why is that? You know, I really like to find protective mechanisms. We talk about things that are risk factors and can cause you to, have hypertension or other, health problems, but rarely do you see something that protects you from something. So we also saw that the C allele was found to be the risk allele here for increases in systolic blood pressure. So this is just a little bit more about the SNP, where it's located on the gene, the mechanism of action, where you can find it in terms of different organs. And so this question was really nagging at me, like, okay, so this T genotype, why is this happening? Why is it protective? And how can we go about unraveling this and making more sense of this? Well, it could be due to other factors, like body mass index or physical activity, which we didn't measure physical activity. So these are other things that could be factors associated with. And then we would have to replicate it in a similar sample that would control for these factors. So, but this small pilot, we did walk away with some conclusions that we could recruit people into the study. People, did trust us to do the work in an ethical way, and we could identify risk factors for hypertension in in young African American girls, even before they expressed the phenotype of hypertension. But it still left us with the limitation, well, can you replicate it? That's always a big question. Any genetic study, can it be replicated? But we were able to publish from this small pilot in various places: Journal of National Black Nurses Association, Biological Research for Nursing, Journal of Transcultural Nursing, Journal of Pediatric Healthcare, and in Journal of Hypertension. But no matter how great you think your study is or how you have unraveled all these mysteries, you always have a reviewer that does not like what you did. And I call this reviewer number 2. So reviewer number 2 said, okay, that's great. You did all this work, you know, but can you replicate this in a similar sample that has that of normal weight African Americans that get a lot of exercise, have a healthy diet, but still have hypertension? And at first you think, well, no, because they probably wouldn't have hypertension if they're normal weight and healthy exercise, and you know they eat a healthy diet. But if you're making the case that genetic risk factors play a role, then you really do have to think about, well, you know, they have a point. So I went and talked to my mentors. I went and talked to James Jackson about this, and he said, you know, you should really talk to an anthropologist. And, you know, so I did exactly what he said. And I went to the dean of the School of Nursing and asked for some additional funds to do a replication study, and also the vice provost here at the University of Michigan to replicate this study in Mali, West Africa. And I'll tell you why Mali, West Africa. These people have lived the same way that they have lived since the early 1400s. They still have problems with hypertension, even though they still have a primitive lifestyle. They still hunt and gather for food. They're mostly underweight instead of overweight. They have a healthy diet, but they still have high blood pressure. And what does that have to do with the people in Detroit? You know, why does that matter? Well, if we think about the transatlantic slave trade, about 40% of the Africans that were transported from West Africa, from the Africans that were transported from West Africa and from the Dogon tribe. And they were transported to slaveholding states here in the Americas. Now, if you think about where people in Detroit come from, you can think about, well, in the early 18 to 1900s, about 90% of all African Americans lived in the South, in slaveholding states. But during the Great Migration, from 1915 to 1930, many African slave descendants relocated from southern states to northern states for better job opportunities. And in particular, Detroit, the big three automakers, would hire African Americans to work in the factories. So from 1910 to 1929, Detroit's black population increased from 6,000 to 120,000, making Detroit a major northern migration destination. So it made it very plausible that there were heritable roots from West Africa to the people that were here in Detroit. And in addition to that, not just the genetic component, this naturally controlled for diet and physical activity and BMI. And this just gives you more images of what the Dogon tribes and the terrain looked like. So it's a developing country, polygamous society. They can only marry within the 7 tribes that are there, so it makes it a very homogeneous genetic population, which if you're doing genetics research, you think this is awesome. And then, but the language in the Dogon tribes was Dogon, but the official language of West Africa is Bambara. So before we even got there, all of our materials had to be translated from English to Bambara, and then we had to hire a translator to orally translate everything to Dogon. So think about IRB application for that. And you're taking DNA from children. Oral, and you're getting oral consent. So that took a long time. But as you can see here, getting to the Dogon tribes, we arrived into the capital of Bamako, and it took about 18 hours by car. There are no roads, so I don't know how drivers know how to get to where they're going. There's the closest hospital was 2 hours away from the Dogon villages. There was one nurse and one physician there. I brought a lot of supplies with me just in case, because I'm a nurse, so I'm always prepared. And I really believe I had more supplies than the hospital had. And then their diet consists of lamb, chicken, fish, lots of vegetables and potatoes. But you don't see any golden arches here. No fast food, none of the stuff that we, find convenient for us here in the Detroit area. And while I was there, I stayed in what was a hotel, but it would not be considered a hotel by any means in the United States. And they had a restaurant, and you just ate whatever they had available. And one day they had chicken, and I said, oh, okay, we'll have chicken. And you could hear them slaughtering the chicken while you were waiting. So after that, I ate a lot of soup because I did not want to hear the chicken being slaughtered. I took a picture of this. This is millet. I didn't know what it was until I went to the Dogon tribes. And it's a grain, and they pound it and mix it with water and drink it. And this is my free research assistant. So how do you get someone to come all the way to West Africa with you, get up at the crack of dawn, carry all your stuff, and label all your DNA tubes and all this stuff? You tell your husband you're going to a polygamous society that can have as many wives as they can afford, and you walk away. And then it's like, oh, I think I'll go with you. That's a great idea. So that's how you get a free research assistant. So as you can imagine, the IRB process for this study was a lot. But, once everything was done, it was great to get there. We hired a translator that could translate all of our materials. We had to meet with the chief of each village, but once the chief gave his permission for us to do the study, then the majority of the women were more than happy to participate in the study. All consent was verbal, and we included 3 generations just like we did in Detroit. In 10 days, we were able to collect data on 199 individuals. And we, in terms of participant honorarium, the maximum we were allowed to give was $1 US, which was equivalent to 500 Central Francs of Africa. So that was considered a lot of money by the Mali government. So each woman was given what was equivalent to $1 US to participate in the study. We collected the samples using the Origin collection tubes because they don't have to be stored by refrigeration. They can withstand the high heat and are easily transportable back to the United States. And we did everything that we did in the Detroit study. The only thing, we started looking at more SNPs because there had been a couple more SNPs that had been identified in the literature as risk factors for hypertension. So instead of 4, we had 6. And we looked at these positional candidate genes associated with hypertension. We used the same statistical analysis plan that we used in the Detroit study. The only difference is that we did not correct for antihypertensive medication use because even if people were diagnosed with hypertension, they weren't taking any antihypertensive medications because the hospital was so far away. And they had other things that they were more concerned about, and rightly so, like malaria and other communicable diseases. These are the demographics. But what I like to point out here is that 35% of the grandmothers had blood pressures that were indicative of hypertension, 10% of the mothers, and 1.5% of the granddaughters. The body mass index, as you can see here, most were either of normal weight or underweight. And when you contrast that with the Detroit study with African Americans, where most people were either overweight or in the obese category. When we did our analysis, our linear mixed modeling, we found one SNP that was significant for predicting increases in systolic blood pressure, and it was the exact same one that we found in the Detroit study. Now, the only thing we didn't find the protective allele that we were looking for. But it was still very interesting that we would find this exact same SNP that was a risk factor for hypertension in both populations, and we were able to identify this in young girls even before they expressed the phenotype of hypertension. So this is the information about the SNP. This was the first time this gene and SNP had been identified in multiple generations of West African Dogon women. And I just said that the results were the same, except the T allele was no longer there. But the important thing about this study was that it provided more evidence that we could identify genetic underpinnings that were risk factors for hypertension even early in life prior to disease expression. We were able to publish in several places, Biological Research for Nursing, Ethnicity and Disease, Yale Journal of Biology and Medicine. Oh, and then, of course, reviewer number 2. It's like, you gotta be kidding me. So you go all the way to West Africa. You replicated this study in Detroit, and you had, you were able to get similar findings. But, 199 people for a genetic study, that's a small sample. Can you replicate this in a large epidemiological sample of African Americans? And at first I thought really defeated. I was like, well, no, I can't because I don't have, millions of dollars to, enroll thousands of people into a genetic study. But then I went and talked to my mentors. And this is why, when you get to unraveling things, you really should reach out and talk to people that have helped you and your resources because they are a wealth of knowledge. So I went and talked to my mentors and other mentors in nursing, and there was a call for a grant, Robert Wood Johnson Foundation Nurse Faculty Scholars, of which Jackie Campbell was one of the people on the National Advisory Committee, Angela McBride, Beverly Malone, all also nursing mentors for me. And I said, if I applied, do you think this would be something that the nurse faculty scholars would be interested in, as a replication study, if I can find a sample with, enough people to see if we can replicate this. So I applied, and I was funded to replicate this. Went back to my mentor at Washington University in St. Louis, Dr. D.C. Rao. I asked him if I could access the Hypergen network, of which they were the data coordinating. Washington University in St. Louis was the data coordinating center for the Hypergen study. And if I was, able to get this funding, could I do this study and access this data set? So he was more than helpful in allowing me to access this data set where I had over 1,200 people that I could analyze the data from. The only difference was that in the Hypergen data set, there were 2 generations, mothers and daughters. All the mothers had been diagnosed with hypertension. All the daughters did not have hypertension. But the other great thing about accessing the Hypergen data set was that they had genome-wide association data. So they had over 1 million SNPs available to interrogate instead of the 4 or 6 that we were looking at in these small pilots. So we went through these controls to actually see how many SNPs we were really going to look at in our analyses. And when we went through these controls, we had a result of 436 SNPs in mothers and 95 SNPs in daughters that we were going to analyze. We used the same statistical methods that we had been using. Here are the demographics. As you can see here, although the mothers had been diagnosed with hypertension, clearly they were taking antihypertensive medication and were being somewhat controlled. And these are the blood pressures, 116/68 for the daughters. Body mass index, these people were recruited from Forsyth County, North Carolina, and in Alabama, I believe. And these were the body mass index that we had, more similar to that of what we saw in Detroit than what we saw in the Dogon tribe. So they were in the obese category, both the mother and the daughters. So what we found when we looked at main effects with SNPs and blood pressure was we expected to find something in the mothers because they all had hypertension. So that was not surprising. But what was interesting was that we found these 6 SNPs that were significant for increases in both systolic and diastolic blood pressure. But none of these individuals have hypertension. So we continued looking at the BMI interaction, and this particular SNP remains significant for predicting increases in systolic and diastolic blood pressure in the daughters that did not have hypertension. So we thought that was very interesting. This particular SNP is located on chromosome 2. It's a different gene than the sodium bicarbonate transport gene that we found in the Dogon and the Detroit sample. But this was the first time that this gene and SNP had been diagnosed, had been identified in undiagnosed African American girls for risk for hypertension. This is the mechanism of action for this SNP. This is where it can be found, but I think we can safely say it was restricted to lung tissue since it was only women in this study. The comparison with the other 2 studies, we found that chromosome 2 was consistently found to be a harbinger for hypertension in Black populations. It was a different gene for the Hypergen study than for the Detroit and Dogon studies. But the consistent results from all 3 of these studies basically showed that we could identify these risk factors for hypertension early in life prior to diagnosis of hypertension, which was really the question we wanted to unravel. Can we answer this question? Can we find these risk factors before people actually develop the disease? And that's the pediatric component of this study. I'm always trying to look for how we can do things to prevent young people from developing these chronic diseases as early as their parents and their grandparents. This is an image of young people that I would bring to Yale. We called it our Ivy League Academy, and from Inkster High School, where I went to high school. And we would bring these young individuals to Yale just to see the types of things that we were doing in science and trying to get them more engaged in science so that to grow the next generation of scientists to continue to unravel all of these questions that we have to try to eliminate some of these health disparities that we have. So what are the implications? Well, if we can identify these risks, it can be a lot easier for us to develop appropriate interventions. We published the results from this study in scientific reports, Biological Research for Nursing. And then I used these studies that I conducted in Detroit, in the Dogon region, and with the Hypergen Network to build for the current R01 that I have, which not only looks at genetic and epigenetic interactions with psychological factors. It looks at not only the genetic and epigenetic component, but also looks at psychological factors that can influence blood pressure. So we're not looking at just the main effects, but we want to know how do these variables work together to influence blood pressure. So some of the psychological factors that we're interested in, and we're using a genome-wide and epigenome-wide approach in this study. We're also looking at ancestry and how that plays a role. But some of the psychological factors that we're looking at are perceived racism and discrimination. We're looking at parenting stress, symptoms of depression, trauma, coping. We interview these women every 6 months, and we follow them for 2 years. So there's 4 interviews. We have completed our enrollment of 500 individuals, 250 families, and we recruited from Head Start programs across Connecticut. So we recruited children ages 3 to 5 and their mothers. Now, I don't say this to say that I'm doing any of this alone. There are more than 13 people on our research team that work to get all of this done. I have a co-PI that is a child psychologist, a co-investigator that is a genetic epidemiologist. I have students, postdocs, undergraduate students, volunteers. If you're more interested in what we're doing with the Intergen study, you can look us up on our website here or Facebook. We have published methods papers if you want to know how to replicate any of our work, because I have learned from reviewer two. So I've published the methods papers. If anybody wants to replicate, this is how you can do it. We've published some of the data from our study in clinical epigenetics. I've also learned from reviewer two, can you replicate this in a large epidemiological sample? Yes, we already replicated in the Jackson Heart study. I don't want to see any of these reviews again from reviewer number two. We've looked at perceived racial discrimination and DNA methylation in the Intergen study. And we've looked at parenting stress and DNA methylation in our sample in Journal of Clinical and Translational Science. So all of this to say, as you continue to unravel, you continue to ask more questions. It's not that you have answered everything. I feel like I have more questions now than when I started. But I feel like we are closer to actually making some change and moving health sciences forward to eliminate health disparities. So the next thing that I'm doing now is I'm looking at, I have a small pilot where we're looking at the genomics of lead exposure in Flint, Michigan. Now, why would I be interested in lead exposure? Well, I'm originally from Michigan, so this really, hit me very hard when I heard about the Flint water crisis, and I don't have to explain it to this group. But it's like, okay, what can you do as a scientist to do anything for this population? Well, I don't know if I mentioned, but during my master's program, we were required to write a master's thesis at Wayne State University. And during that time, my advisor and I, we worked on this study where we looked at allelic variation in environmental lead exposure in African American children in Detroit. And the reason why is because I had that mobile clinic, and I would have kids that came into the clinic with signs and symptoms of lead poisoning, but I would check their lead levels, and their lead levels would be below the CDC guidelines for treatment. So their lead levels would be 5 micrograms per deciliter or lower, which again caused me to have more questions like, why do they have these signs and symptoms if their lead levels are normal? Well, I continued to read and unravel more, and I ran into the work of Dr. Ron Peretz and his group, where they had identified the ARSA gene, that if you are heterozygous for this particular gene, that you can have the neurotoxic effects that you see in people that are that have high levels of lead exposure, even at low levels of lead exposure. Causes the same demyelinization of the neurons as you see with lead poisoning. And who is at greatest risk to be heterozygous for this gene? People of African ancestry. About 42% of people about 42% of the people in my study, my master's thesis, were carriers, were heterozygous for this particular gene. When you are heterozygous for this, you have a condition called aryl sulfatase A pseudodeficiency, which is when you have this demyelinization of the neurons. How do you treat it? You treat it the same way that you treat people that are lead poisoned. You give them iron supplements, green leafy vegetables. So it seems like a very simple treatment. If you're homozygous for this particular gene, then you have a condition called metachromatic leukodystrophy, and these children usually die early in infancy. So I was very upset to learn that although children, and I had published this, and other people have published on this topic, so I was very upset to learn that although children were being tested for lead poisoning in Flint, if their lead levels were below 5 micrograms per deciliter, there was no more follow-up required, which we know this is not the correct way to treat these individuals based on science. So I wrote a scathing commentary to MPJ Genomic Medicine saying what I thought needed to be done and how the CDC guidelines need to take into account the precision health perspective that we're talking about in terms of precision medicine and the PMI initiative and so on and so forth, and how this totally ignores precision health, and how people need to be tested and treated. So later that year, I was awarded the Presidential Early Career Award from President Obama. So I'm continuing working with people, Cleo Caldwell here in the School of Public Health, to work on this particular project in Flint. Additional things that I'm doing, President Obama awarded the PCASE award. With that comes additional funding to further explore more questions that are related to your R01 that you currently are not exploring. So what I'm doing there is looking at using whole genome sequencing as an educational tool in the African American community. And why is that important? Well, first of all, I think it's important to illustrate how nurses are important in precision health and genome sequencing, that we are pivotal in not only ordering the tests, analyzing the samples, and analyzing the data, revealing the results to the patient, and conducting the genetic counseling that needs to be done. So I thought people needed to understand this, so I wrote an article about the genome sequencing and the importance of it. But then I also felt like it was important for people to understand based on an example. So if you think about familial hypercholesterolemia, people that have familial cholesterolemia typically don't know they have it until they have a sudden cardiac event. Many African Americans have died from sudden cardiac death. Typically, you're not tested for this until after the fact, unless you can afford to pay $2,000 to have your whole genome sequence done by Illumina or whatever other company that you can have your whole genome sequence analyzed. But if this is something that we can do in the community, just like we can do any other genetic test, and we can identify people that have this variant for familial hypercholesterolemia, we can put these people on statins like Lipitor, and they won't die. So I think that's a very good reason to do whole genome sequencing in the community, particularly in minority populations that are underserved when it comes to whole genome sequencing. So getting to the last part of unraveling health disparities for African American women, I think it's not only important to do your job as a scientist or to do my job as a scientist, but to help train other people so that we can keep the momentum going and continue the work ahead. So I always like to talk about my mentees and postdocs. And I talked to Ada Sue Henshaw about this the other day. It's just a coincidence all my postdocs are from Michigan. Coincidence. Michelle Wright, she was a postdoc in my lab where she worked on the Intergen study. We did a lot of work there. She's interested in maternal child health. She published more than 10 articles while she was a postdoc with me. She is now faculty at University of Texas, Austin, where she recently received a K Award. So I feel like, you know, when I was here at the University of Michigan as a junior faculty, people really helped me along in my career and made sure that I was able to get the funding and the resources that I need to be successful. And I try to do that with my mentees so that they can continue to do the important work that needs to be done. Veronica Barcelona de Mendoza, who is a graduate of University of Michigan School of Nursing undergrad program. She was a postdoc in my group, and she's now assistant professor at Yale School of Nursing. She also, I'm her primary mentor on her K Award, where she's looking at DNA methylation and preterm birth in African American women. And as we know, that is a huge problem with birth disparities among African Americans. She's also been very successful. She was a Jonas Policy Scholar because when we think about health disparities, it really does go to the point where policy change is necessary. So being a Jonas Policy Scholar, she was able to work with the genetics expert panel with the American Academy of Nursing and write policy briefs and talk about the changes that needed to occur to address health disparities in minority populations. I was also fortunate enough to be able to run a summer intensive where we invited 7 diverse scholars from across the country to participate in a health disparity summer intensive where we talked about career development. We helped pair these junior faculty or postdocs with mentors. And Gandhi, I think, said, got this right even back then, especially in today's society where we have Twitter and Facebook and so on, that a sign of a good leader is not how many followers you have, but how many leaders you create. So here's an image of the summer intensive that I held at Yale in my role as Associate Dean of Diversity and Inclusion. So that's Veronica, which I've already talked about, Marie Ann, who is actually faculty here in University of Michigan, and she is now an administrative role. Paulie Joseph, who is a tenure track at the National Institute of Nursing Research, doing research in genomics and taste sensory genes. Dr. Billy Caceres, who is now a postdoc at Columbia University and also working with me on the Intergen study where he's looking at trauma and DNA methylation. Jasmine, she is now at Yale University, part of the National Clinician Scholars Program. Tamaya Nolan, who is now assistant professor at Ohio State University. And Kia Skrine, who is at UCLA in their National Clinician Scholars Program. This is Dori Dumas, who is the president of the local New Haven chapter of NAACP. I brought her in to talk to our group because I think it's very important if you're doing work that's in the community to be very closely knit with community organizations and understanding what their values are and how they can help you to also work with the community that you're trying to reach. So again, I wrote an article on omics-based research and precision health and what are some of the things we need to do to improve in this area. And as I write these things, I'm actually doing all the things that I'm recommending. So as soon as I arrived to NYU, we wrote a P20 Center grant that was recently funded on precision health and diverse populations. I'm a co-PI with Dr. Gail Melkis. And I think it really, when you talk about unraveling, that this has really brought it full circle. I was a recipient of a P20 pilot in my very first grant, and now I can help other people to receive these pilot grants so that they can continue to build their careers and do the things that are important to unravel health disparities. Our center focuses on multiple chronic conditions and diverse adult populations. So unraveling is just not one thing at one time. I think you can unravel or continue to answer questions in many ways. I do so not only with my individual research, with the PCAS Award, but also with other grants to help other people to look at answering these questions and addressing health disparities. Also as a mentor to K Awards and other career development awards, and even through foundation and other small grants. So I know I'm 5 minutes over time, so I want to thank you for your time and attention and for having me here. It's always a pleasure to be back in Ann Arbor at the University of Michigan. And I hope that you learned something from this talk and that you can continue to unravel these questions we have to address health disparities in the future. Thank you so much.
If you want to stay around later and ask questions, please feel free. As you leave, make sure you fill out the survey. We will be pulling the raffle in a few minutes. We have a gift for you. As our keynote speaker, you want to present it, Guy?
Keep talking. No, you. You talk, I'll present. Here we are.
So on behalf of the health science schools, colleges, and schools, colleges, and units, and our deans, we thank you so much for granting us your lecture and for being with us on this day. Let's give Dr. Taylor a big hand. Thank you. So there's plenty of food out after the reception. Dr. Taylor will be around for a little while. Complete your surveys, and we're going to start calling. Dan and Ethan are back there. We're going to start calling names and pulling out for the gifts. Thank you so much.
Thank you for having me. I hope it's what you
Oh, excellent.